Hello and welcome to my first YouTube tutorial, and in this tutorial I'll be covering the Biology Module 1, which is called Key Concepts in Biology. So all this information you will be asked questions about on both Paper 1 and Paper 2 in your final exams. So the first thing I want to talk about are cells. So there's animal cells, plant cells and bacterial cells that you need to know about. And in each cell there are smaller parts of the cell which do specific jobs. So first of all, you need to know about the nucleus. The nucleus is a part of the cell which contains the chromosomes, which are made of DNA, and this controls the function or the activities of the cell. The cell membrane is the outer part of the cell, and this controls what passes in and out of the cell. So the substances that go in and out of the cell are controlled by the cell membrane, and the cell membrane also separates one cell from another. The mitochondria is a very small structures, and they look a little bit like a jelly bean, and the mitochondria are responsible for aerobic respiration. The ribosomes again are extremely small structures, and they can only be seen with an electron microscope, and the ribosomes are responsible for making new proteins. So plant cells have all the components that animal cells have, but in addition they also have a cell wall, which is the outer part of the cell, and it supports the cell. It's made of cellulose, which in your diet you would class as fibre. You've also got the chloroplasts, which are the part of the plant cell which contains the chlorophyll, which is responsible for photosynthesis, and the other additional part is the vacuole, which is filled with cell sap, which is sort of a watery substance, and that helps to keep the cell nice and firm and rigid. The bacterial cells are a little different from plant cells and animal cells in that they don't have a nucleus, so their genetic material can be in the form of either chromosomal DNA or plasmid DNA. Bacterial cells also have cell membranes and ribosomes, and additionally they have a structure, or some bacterial cells have a structure called a flagella, and that is responsible for propelling or moving the bacterial cells. So it's essential when you're revising that you are able to recognise the parts of the cell and be able to describe what each part of the cell does. So I'm going to annotate this diagram, and then I'm going to delete all the annotations. It might be a good idea if you copy this diagram down and try and do it by yourselves. So you can see the diagram is now annotated. So the first thing that you need to know is the nucleus, which is in the middle of the cell, and that's where all the genetic material is. Then on the outside of the cell, around the outside here, you've got the membrane. The main part of the cell, sort of this blue stuff here, is called the cytoplasm. That's basically where everything just floats around inside there. And then you've got the mitochondria, which you'll have lots of, especially in cells that require lots of energy. Obviously that's where aerobic respiration takes place, where energy is released. And then you've got these really small structures, the ribosomes, and that's where the proteins are made. So I'm going to delete the annotations off the slide, and then I would like you to pause the video and see if you can re-annotate and explain what each part of the cell does. So we're going to do the same again with this slide. So this is obviously a plant cell. And so I'm going to annotate the cell again and explain a little bit about what each part does, and then clear them off and expect you to pause the video and see if you can annotate a diagram or draw your own little sketch of a plant cell and annotate it and explain to yourself what each part does. So as you can see, the cell is now annotated. I'm just going to go through each little bit to reiterate what each part does. So the top one is the mitochondria, and again exactly the same in a plant cell. This is where aerobic respiration takes place. The cell wall is the outermost layer of the outside of the plant cell, and that is what keeps the plant cell rigid. So this is the cell wall on the outside, and then you've got the cell membrane, which is sort of the inner part, and that controls what goes in and out of the cell. In the middle here, you've got the vacuole, and that is filled with the cell sap, which keeps the plant cell nice and rigid. And then we've got the nucleus, which contains the genetic information, the DNA, and the chromosomes, which are made of DNA. The cytoplasm, again, just fills the cell. And the ribosomes, these small structures down the bottom, and that's where the plant will make its proteins. Okay, so I'm going to pause the video now and delete the annotations, and then you can try and do it yourself. Okay, so the final cell we're going to look at is a bacterial cell. So exactly the same process again. I'm going to annotate the cell, explain what each part does, and then you can try and do the same process yourself when the video is paused, when I've deleted the annotations. As you can see, I've annotated each part of the bacterial cell. Okay, this first part is the slime coat, which is on the outside of the bacterial cell. Now, not all bacteria have this, but it is for protection of the bacterial cell. The cell wall, which is just around the outside here, that is not made of the same substance as in plants, so it's not made of cellulose, and it's quite a flexible cell wall. And then we've got the cell membrane, sort of the innermost layer of the outermost part of the bacterial cell. And the cell membrane does the same function as in plant and animal cells. It controls the substances that pass in and out of the bacterial cells. And then we've got the different types of DNA. So we've got the chromosomal DNA, which is the sort of oddly shaped part in the middle, and the plasmid DNA, which is always quite circular. And the final part is the cytoplasm, which is just the same as in animal and plant cells. It's just the substance where everything else is floating around inside the cell. Okay, so I'm going to remove the annotations now, and then you can pause the video and try and draw a bacterial cell and annotate and explain what each part of the cell does. In addition to knowing the different types of cells and what the different types of cell organelles are and what they do, you also need to know some things about some specialized cells, which are basically cells which do a very specific or special job. So the first cell I'm going to talk about is the sperm cell, and this has quite a few quite unique features which allow it to do its job. Right at the front of the sperm cell, the head of the cell, is something called the acrosome, and this contains an enzyme which helps break down the substances in the outer coat of the egg cell, which basically allows the sperm cell to get into the egg cell so that the nucleus can deliver the genetic material. The nucleus is a little bit different than in a normal cell. The nucleus is called a haploid nucleus, which basically means it's got half the number of chromosomes, and that will make sense a little later when I explain about the egg cell. Now, sperm cells have a lot of mitochondria, so you can have a think about why that is. You can pause the video if you like. Okay, so the mitochondria are the part of the cell organelles that release energy. So sperm cells need a lot of energy because they need to swim a relatively long distance to get to the egg cell. The final part of the sperm cell you need to know about is the tail. So it's got quite an obvious function. The tail moves from side to side, allowing the sperm cell to move as they travel towards the egg cell. So the second specialized cell that you need to know about is the egg cell. So this is much bigger than the sperm cell. As you can see, these images are not to scale. The sperms are much smaller than egg cells. In fact, the egg cell is the largest cell in the human body. It's about the size of a full stop you would make on a piece of paper. So around the outside of the egg cell, we've got a substance called a jelly coat, which protects the cell, and it also hardens after the cell has been fertilized to prevent any further sperm from entering the egg cell. The nucleus of the egg cell is also a haploid, which again means it's got half the number of chromosomes. So basically, in humans, we have 23 chromosomes in the egg cell and 23 chromosomes in the sperm cell. And when these two combine, in normal cells, which are called diploid cells, we have 46 chromosomes. And the final thing you need to know about the egg cell is the cytoplasm, which is the main part of the cell. It's packed with nutrients, so it's providing the egg cell with enough energy when it has to grow and develop into an embryo. And the final type of specialised cell that you need to know about are the ciliated epithelial cells. So ciliated epithelial cells have the structure called cilia on the outside of the cell, and they're usually found in places where something needs to be transported. So, for example, in the lining of the oviducts, which are sometimes called the fallopian tubes, they will have ciliated epithelial cells which will transport the egg along the tubes on its journey to the uterus. So here are 5 review questions for you to attempt. I would suggest that you pause the video and write your answers down, and then I will go over each question, explaining the question and giving you the answers. Okay, so the first question is, name the cell organelles that are present in both animal and plant cells. So the common subcellular structures that are found in both animal and plant cells are the membrane, the cytoplasm and the nucleus. However, the organelles are the small parts of the cell which are contained in the cytoplasm, and they will be the ribosomes and mitochondria. Question number 2, describe the genetic material in bacterial cells. So the genetic material in bacterial cells isn't contained within a nucleus, and it comes in 2 different forms. There's the chromosomal DNA and the plasmid DNA. Question number 3, what is the function of a sperm cell's acrosome? Well, the acrosome is at the tip of the head of the sperm cell, and it contains an enzyme which will break down the outer coat of the cell, the egg cell, to allow the sperm cell to penetrate inside of the egg cell for fertilization. Question number 4, why are both sperm and egg cells described as haploid? And this is simply because they have half the number of chromosomes than a normal cell. So in humans, both sperm and egg cells only have 23 chromosomes, whereas a normal cell, which is described as diploid, has 46 chromosomes. And the final question, question 5, describe the function of ciliated epithelial cells. And that is simply they are involved in moving or sweeping substances along, usually tubes, inside the body. So the next topic we're going to cover in this tutorial is about microscopes. So hopefully you'll all have done a practical involving microscopes, and you might have prepared a slide to look at onion cells, and you might have even prepared a slide to look at your own cheek cells. So the 2 main parts of the microscope that you really need to know about are the 2 different lenses. So at the top here, you've got the eyepiece lens, and at the bottom, nearer the object you're looking at, you've got the objective lenses. And the way you can calculate how much magnification a microscope has is you multiply the power of the eyepiece lens, or the magnification of the eyepiece lens, by the magnification of the objective lens. So for example, if the eyepiece lens has a times 10 magnification, and the objective lens has a times 20 magnification, you simply do 10 times 20, which will give an overall magnification of times 200. Other parts of the microscope that you might be expected to know will be this stage, which is this bit here, and you also have a light, which is projecting light through there. You've also got the focusing wheels, which are here. One of the most difficult things they can ask you about magnification is working out the actual size, or the image size, or the level of magnification from a microscope. So this is one of the equations that you need to remember for biology, and I always remember it as AIM. So you've got the A, which stands for the actual size, the I, which stands for the image size, so the size of the image that you're looking at, and the M, which stands for the level of magnification. So if you had an image, a photograph, or you measured it through your microscope and it was 10 millimeters, but you knew the actual size of the thing you're looking at was 100 micrometers, you will be able to work out the magnification. So I'll let you have a bit of a pause of the video and see if you can do that by yourself, and I'll explain what you should have done in a second. Okay, so the first thing you need to do is make sure that both the image and the actual size are in the same unit. So if the image was 10 millimeters and the actual was 100 micrometers, that is actually 0.100 millimeters. So now all you need to do is divide the image by the actual to give you the magnification. Okay, so when you divide 10 by 0.1, you're going to get a magnification of times 100. So this is just a bit of a reminder of the different sizes of images, or actual sizes that structures you're looking under a microscope could be. So the first one, milli, basically means 1,000th of a meter. So there are 1,000 millimeters in 1 meter. And that can be represented in the standard form 1 times 10 to the minus 3, which means a 1 preceded by 3 zeros. Okay, micro literally means a millionth of a meter, or a millionth of a unit, and the symbol is a Greek letter. It's written like that. And that literally means it's a 1 preceded by 6 zeros and can be written in the standard form. Okay, the final two are the nanometer and the picometer. So a nanometer is a billionth of a meter. And a picometer is a trillionth of a meter, but you'll rarely be asked about the picometer. So as you can probably see, a millimeter is 1,000th of a meter. A micrometer is 1,000 times smaller than a millimeter. A nanometer is 1,000 times smaller than a micrometer, and a picometer is 1,000 times smaller than a nanometer. And obviously you can go in the opposite direction. So a nanometer is 1,000 times bigger than a picometer. A micrometer is 1,000 times bigger than a nanometer, and a millimeter is 1,000 times bigger than a micrometer. Or you could say there are 1,000 micrometers in a millimeter, just as there are 1,000 millimeters in a meter. Okay, so another common question they like to ask about in exams is about the resolution of a microscope. And basically, you need to know that an electron microscope has a much greater resolution than a light microscope. And that basically just means that you can see objects inside the cells in more detail through an electron microscope. So as you can see in this diagram, this is through a light microscope, and you can see several plant cells there, and average plant cell or palisade cell inside a plant is about 10 micrometers in length. But in the diagram below, you can see the scale is much smaller. That's 1 micrometer. So you can see much closer up in the cell, and you can see the different parts of the cell. So you can see the cell organelles inside the cells. And it basically means resolution, basically means it's the smallest distance between 2 points that can still be seen as 2 points. So electron microscope, you can see much smaller things in more detail than you can through a light microscope. Okay, so here's some more review questions. I suggest that you pause the video and attempt to do the questions yourself, and then I will go through the answers and explain a little bit more about each question. Okay, so question 1. All you need to do here is simply multiply 10 by 40, and that obviously equals 400. Number 2, if the actual size of a cell is 7 micrometers and it appears to be 4 millimeters as an image on a photograph, by how much has the cell been magnified? So what I would do here is I would draw my triangle, so AIM, and what I've been given in the question is the actual size, which is 7 micrometers, and I've also been given the image size, which is 4 millimeters. So the next thing you need to do is convert your micrometers into millimeters by dividing 7 by 1000, which gives you 0.007, and then you simply do 4 divided by 0.007, which gives you an answer to 3 significant figures of a magnification of 571. Question 3 is convert 3 micrometers into nanometers. So a micrometer is 1,000 times bigger than a nanometer. So to convert it, you just need to do 3 times by 1,000, which obviously equals 3,000 nanometers. Question 4, if one image has a better resolution than another, what does this mean? And that just simply means you'll be able to see more detail with the microscope which has the better resolution. So for example, you'll be able to see the details of the smaller organelles with a microscope with better resolution, and electron microscopes have a much better resolution than a light microscope. And question 5, explain why electron microscopy was needed in order to gain a better understanding of mitochondria, and that is all to do with the resolution. A light microscope, you might just be able to see the mitochondria, but you'll be able to see absolutely no detail. So with an electron microscope, scientists were able to see the details of what the mitochondria looked like and an idea of how the structure was helping respiration. So moving on to enzymes now. So basically you can think of enzymes as a biological catalyst, and what that means is that they speed up reactions in the body. So they speed up processes that would happen, but would happen much slower without the presence of enzymes. So the food that we eat is in the form of large molecules, sometimes referred to as macromolecules, and these need to be broken down into smaller pieces before they can be absorbed by the body. So any protein that we eat, so things like meat, fish, eggs, have lots of protein in. The protein will be a large molecule, and that won't be able to pass from the gut into the blood. So we have enzymes in our body, in our digestive system, that break down the bonds between the individual monomers of the protein, and those monomers, which are single small molecules, are called amino acids. So we can say that protein is broken down by enzymes into amino acids. Other food groups that we eat are things like starch, which are basically carbohydrates, are also large molecules. So the enzymes that break down starch are called amylase enzymes or carbohydrate enzymes, and again it's the same process. You're breaking down the bonds in between the molecules to form smaller monomer molecules, and the monomers that make up the starch are called glucose molecules. So in summary, when you eat a piece of bread or potato, you will break down the starch molecule into smaller glucose molecules, which you can then pass into the blood because they are soluble in the liquids in your gut. And the final type of food group that you need to know about are lipids, which you probably know as fats, but they can be fat as in solid fat, which you usually find in animal products, or they can be liquid fats like oils, which usually come from vegetable sources. And again, lipid molecules are large molecules, and they are broken down into the monomers, and this time there are 2 different types of monomer. There is the fatty acids and the glycerol. So you can see there are 3 fatty acids attached to every glycerol, and the enzymes, which are called lipases, break down those lipids into the fatty acids, which can then be transported across the gut into the blood and used for energy. So the enzymes that do these jobs they're just a type of protein, and they break down the large molecules into smaller molecules, or they can actually build up small molecules into larger molecules. And they do that by bringing together the substrates, which are the things that are being made together or broken apart, and they bring together the substrates in what's called the active site. Now the active site is a specific shape, so a lot of scientists use an analogy to say that the substrate fits into the active site like a lock fits into a key. So it's called the lock and key theory. Now, enzymes are a type of protein, and all proteins are sensitive to changes in temperature. So if the temperature gets above what's called the optimum, which is the most suitable temperature for the enzyme's active site, which is shown here, will be denatured, which means changes shape, which is shown here. And that means that the substrate will no longer fit into the active site of the denatured enzyme. So that means that the denatured enzyme will no longer be able to do its job. So it won't be able to break down the starch into glucose, or it won't be able to build up amino acids into new proteins, because enzymes can do both things. They can break things up and build new molecules. So often in exams, you'll be asked to identify the optimum temperatures or optimum pHs for an enzyme. And so you can see on this graph here, along the x-axis, you've got the pH, and on the y-axis, you've got the rate of reaction. So as you can see, the optimum pH for this enzyme is somewhere along the middle of the scale. So that could be pH 7. It depends on what the scale is. Obviously, there's no scale on this diagram. So you can see that the peak of the graph, you link that down to the x-axis to what the optimum pH would be. In an exam, you might be asked to describe the different parts of the graph. This graph at the bottom is showing you what happens as you increase the substrate concentration. So the substrate concentration is the amount of things that are meant to be reacting together. So as you can see at the beginning of the graph, the rate of reaction increases rapidly, but after a while the line goes flat. So I want you to pause the video and think about why the line goes flat even though the substrate concentration is increasing. Okay, so the reason that the line goes flat is because although the substrate concentration is increasing, there is a limited number of enzymes. Okay, so once all the enzymes' active sites are filled, then the reaction can't get any faster. So no matter how many more substrates you add into the concentration, there's only a limited number of enzymes, and therefore the rate of reaction can't get any faster because the active sites are all full, or they're all being filled as soon as they've done their job. Okay, so the final part of the key concepts and the foundation biology is the transport of substances, and this basically covers how different substances are transported, usually across membranes, but sometimes in the case of plants, it's from the outside of the leaf to the inside of the leaf, or the inside of the leaf to the outside atmosphere. So the easiest method of transport to understand is diffusion. Diffusion is basically the movement of particles from where they are most concentrated. So you can see here, there are far more little green circles in that part of the diagram than there are over here. So that means that this part of the diagram, there's a higher concentration than there is in that part of the diagram. So what would actually happen is, in this part of the diagram, these particles will be colliding with each other quite often, and that means they will be spreading out quite quickly. As they collide, they will move away from each other, And that means that as they move away from each other, they become more spread out, and therefore they will collide with each other less. So the rate of diffusion is always highest where there is a higher concentration. And the reason that diffusion slows down when there is a lower concentration is purely because the particles will be moving around and colliding less frequently than they are in the higher concentration. So common examples of diffusion would be simply smells spreading out from their source where they're most concentrated. So if you imagine someone's baking a cake and you're in a different room, the smell of the cake will be strongest in the kitchen where the oven is, and it will spread around the house by a diffusion. But as you get further away from the source, you are less likely to be able to smell the cake because the particles that are causing the smell have spread out too much for you to be able to sense them in your nose. Other examples would be when you apply perfume or aftershave to your skin. The most concentrated area of perfume or aftershave is on your skin, and it diffuses out into the room, and people in the room can smell what aftershave or perfume you're wearing because the particles are diffusing from your skin and spreading out. But the strongest smell will be nearest your skin. Now, a very specific type of diffusion is osmosis. Now, osmosis is, in simple terms, it is just the diffusion of water. So there are two ways of thinking about osmosis. You can either think of it as the diffusion of water from where there are more water molecules, i.e. on the Y side of the membrane here, to where there are fewer water molecules, which are on the X side. So I should say as well that osmosis has to take place over what's called a semi-permeable membrane, which means that some things can get through the membrane but other things can't. But water is quite a small molecule, so water can usually pass through membranes with small holes in them. Now, the other way to think about osmosis is that the water will move from where it is most pure. So on this side here, there are fewer molecules dissolved in the water. You can see these yellow things are molecules that are dissolved in the water, and they will move to an area where there are more things dissolved in the water. As you can see, there's more little yellow things here. So water will always move from where it is more pure, which is here, to where it is less pure, which is over there. So that means, for example, if you were to put a piece of potato into some water, so here's a piece of potato, the potato will have different things dissolved inside its cells. And if you put it into a beaker of pure water, the water will move. So there'll be some water inside the potato as well, and some water outside the potato. But because the water outside the potato is more pure, or there's a higher concentration of water molecules, the water will move in to the potato cell, making the potato cell gain mass. Now, this is often a question that is asked on the exam paper, and you do need to be able to explain osmosis in terms of moving into potatoes, but really it is about moving into cells of any kind. Now, the final type of transport that you need to know about, so this is again reminding you that it's transport across cell membranes. So you can see in this diagram it does actually show you a cell membrane. And this is a type of transport that actually requires energy. So with diffusion is the movement of particles down what's called a concentration gradient. Okay, so imagine if you put a ball at the top of a ramp, the ball would naturally roll down the ramp. However, active transport is for when there is a low concentration of molecules on one side of a cell membrane, and the body or the plant wants to get the low concentration into the cells where there is actually a higher concentration of those molecules. So active transport goes against the concentration gradient, which is a little bit like a ball moving up a hill. So you might be thinking, well, a ball wouldn't move up a hill by itself, and that's exactly what happens in active transport. Active transport can't happen without the addition of energy from the organism. So active transport requires energy to move substrates or move molecules against a concentration gradient. So an example of this would be the movement of nitrates from soil into the root hair cells. So the plant will take the nitrates because it knows, or it's evolved to grow better if it takes advantage of all the nitrates available in the soil. So it will move the nitrates from the soil into the root hair cells where there might already be a high concentration of nitrates, but the plant is prepared to sacrifice some energy to transport the nitrates from one side of the membrane, which is the soil, into the cell cytoplasm inside the cell across the cell membrane and cell wall. Okay, so here are the 5 review questions on the topics we've just done on enzymes and on transport of substances. As usual, just pause the video, answer the questions, and then I'll give you the answers and explanations in a few seconds. Okay, so question 1. An enzyme called pepsin can break down the proteins in our food. What products are made available by this process? So pepsin is the enzyme, and that will break the bonds down in the protein, and proteins are made of a monomer, which is a small molecule called amino acids. Okay, number 2, explain why having a high body temperature is very dangerous for our health. Okay, so this is all to do with the enzymes in our bodies being sensitive to temperature. So basically our body temperature should be above 37 degrees C, but definitely below 38 degrees C. If our body temperature goes above 40 degrees C, we can be in real danger. And this is because the enzymes which are vital for keeping us alive will become denatured. And that means the active site of the enzyme changes shape, so the substrates will no longer fit in the active site, so the reactions won't happen, and therefore we might our cells might not be able to get any energy from respiration, and therefore the cell will die. Number 3, explain why enzymes that work in the stomach do not work in the small intestine. And this is all to do with pH. So enzymes are also sensitive to pH as well as temperature, and specific enzymes only work at specific pHs. So the stomach, as you might know, is quite acidic, but the small intestine has an alkaline environment. So the enzymes that work in the stomach do not work in the small intestine because they've been denatured. Number 4, explain diffusion and state why osmosis is sometimes referred to as the diffusion of water. Okay, so diffusion is simply the spreading out of particles from where they are more concentrated to where they are less concentrated, and this requires no energy. Scientists call this passive. Osmosis is sometimes referred to as a diffusion of water because in its simple terms it is the movement of water from where there are more water molecules to where across a semi-permeable membrane to where there are fewer water molecules. Alternatively, you can say it's a movement of water from where there are more things dissolved in the water, so where there is things with higher concentration, to where there is a lower concentration of substrates. And again, it has to be across a semi-permeable membrane. And question 5. Explain why plants have to rely on active transport to absorb some minerals from the soil into their root hair cells. So this is basically because the plant wants to absorb as many minerals from the soil as it can. So rather than allowing the minerals just to diffuse into the root hair cell, so from where there's a high concentration in the soil into a lower concentration in the cell, eventually those two concentrations would balance and there'd be lots of nutrients left in the soil. So the plant uses active transport, which is using energy to transport the minerals from the soil from a low concentration into a high concentration in the cells. So it's basically just so the plant can absorb as many minerals from the soil as possible. Okay, so thanks for watching. Hopefully you've learned a bit, or you've at least reviewed what you've been doing. There'll also be some more questions available, and we'll put those in the links under the YouTube video. Good luck.